welcome back to my channel. In the last two parts of the video, we have discussed in detail about the digestion in the alimentary canal and in the how the associated glands helps in the alimentary canal to digest. In this part of the video, we are going to discuss particularly how the digestion of carbohydrate, protein and fats takes place. If you have not watched the last two parts of the video, please do watch and comment. So, let us start the topic. First, we will discuss about the digestion of carbohydrate. So, the majority of the carbohydrate which we intake are in the form of sucrose, lactose and starches, and rest of the carbohydrate which we intake is in the less amount. As we have discussed earlier that the digestion process, its main motive is to break down complex substance into simpler form, and the cellulose also we intake in our diet. The main purpose of the digestion is to break down complex into simpler form, and the food which we intake is always in a complex form. So, the carbohydrate which we intake is in the form of polysaccharide, and let us say starch, which is then converted into disaccharide, that is maltose, and then into monosaccharide into more simpler form, that is the glucose. If you do not know what is polysaccharide, disaccharide and monosaccharide, they are basically the types of carbohydrate. Monosaccharide are the simpler form of carbohydrate, let us say glucose is our daily intake we know. Many monosaccharide forms the disaccharide and many disaccharide forms the polysaccharide, let us say starch, which we take in our diet. So, these are then digested into polysaccharide, disaccharide and then monosaccharide. How does this happen? Let us see. In the last part of the video, we have seen that how the associated glands secrete certain enzyme, and now we will see what they actually do. So, the polysaccharides, let us say starch, they are digested into disaccharide form, that is maltose, with the help of enzyme, that is ptyalin, which is secreted from the saliva, as well as with the help of pancreatic amylase, which is secreted from the small intestine. So, both of them help in the digestion of polysaccharide into disaccharide. Now, once they are converted into disaccharide, that is maltose, say, with the help of maltase and alpha dextrinase, they are converted into monosaccharides, let us say glucose, and now the glucose is the simpler form which can easily be absorbed in our alimentary canal. Now, for second example, we can say the lactose, which is a disaccharide, is converted into galactose by the enzyme, that is lactase enzyme, secreted from the small intestine. Next, we will see what is the role or how the digestion takes place in the mouth, then in the pancreas or in the intestine, we will discuss now. So, mainly the carbohydrate digestion takes place in the mouth, because the mouth contains the enzyme that is known as the ptyalin, and ptyalin is an alpha amylase. And what is the role of amylase enzyme? Basically, it breaks down the carbohydrate into maltose and finally into glucose. So, the ptyalin enzyme, which is alpha amylase, which is mainly secreted from the parotid gland. What is parotid gland? We have discussed in the last video. So, ptyalin hydrolyzes starch into maltose and finally into glucose. And very important point to be noted that the alpha salivary amylase mainly is activated at a pH 6.8, not more than that, not less than that. The optimum pH is 6.8, and as the food remains in our mouth for a very short period of time, so only 3 to 4% mainly is digested in the mouth. Next, coming to the stomach part. As the stomach part, we know that it secretes the gastric juice, and gastric juice contain HCl, and the HCl destroys the ptyalin enzyme, which is responsible for the breakdown of carbohydrate. So, no digestion takes place in the stomach region. Next, coming to the intestine, which secretes pancreatic amylase, and it also get activated at the pH that is 8.8. and the small intestine secrete 4 types of enzyme, that is maltase enzyme, sucrase enzyme, alpha dextrinase enzyme and lactase enzyme. What does they do? Mainly maltase enzyme convert maltose into 2 molecule of glucose, sucrase converts sucrose into glucose and fructose, and alpha dextrinase convert limit dextrin into glucose and lactase into lactose and glucose and galactose. So, in short we can say they mainly convert the disaccharide into monosaccharide form. So, here you can see that how the digestion of carbohydrate took place. Moving to the next part, that is digestion of protein. What are protein? Mainly they are the long chain of amino acid which are linked by the peptide bond, and the digestion of protein mainly takes place by the digesting enzyme, that is protease. So, there are 2 types of peptidase, mainly endopeptidase and exopeptidase. As the name suggests, endopeptidase cleaves the internal peptide bond, example is pepsin and trypsin, and exopeptidase is a cleaves the terminal bond, example is carboxypeptidase and aminopeptidase. What does they do? Mainly the pepsin and trypsin is responsible for breakdown of protein into smaller form, and carboxypeptidase acts on the carboxyl terminal of the amino acid and the aminopeptidase on the amino part. So, what happens? So, the protein mainly by the endopeptidase, mainly that is pepsin and trypsin, is converted into polypeptide, and polypeptide, that is by the help of exopeptidase, that is carboxypeptidase and aminopeptidase, is converted into dipeptide, and dipeptidase with the help of dipeptidase enzyme, it is converted finally into amino acid, that is the simpler form. Now, we will see where does the digestion of protein takes place. So, no digestion of protein takes place in the mouth region as there is no digesting enzyme. Second, stomach contain HCl and pepsinogen, as we have discussed in the last video, that the gastric juice secretes HCl and pepsinogen, which is responsible for breakdown of pepsinogen is converted into pepsin. And also the intestinal secretion, that is pancreatic protease, secrete enzyme. Mainly they are secreted in the inactivated form, that is in the form of trypsinogen, chymotrypsinogen and procarboxypeptidase, which are in the inactivated form. Enterokinase convert them into activated form. So, what is the role of trypsin? Trypsin mainly help in the breakdown of protein into peptide, and they do not act in the milk protein. Important point. Chymotrypsin mainly act on the milk protein, that is casein, into paracasein. Carboxypeptidase, they convert smaller peptide into amino acid. In short, we can say they make them more simpler. And intestinal juices also secrete certain enzyme, that is enterokinase, carboxypeptidase and aminopeptidase, and the role of all three I have discussed already. So, we have seen that how the digestion of protein takes place mainly in the stomach and the intestine region. No digestion of protein takes place in the mouth. Moving to the last part of our digestion, that is the fat. Fat mainly digestion into emulsified fat with the help of bile, which is secreted from the liver, and I have already discussed in the previous video. And pancreatic lipase also helps in the digestion of this emulsified fat into more simpler form, that is fatty acid and glycerol. An interesting part is that there is no digestion takes place in the stomach as well as mouth. Very less digestion takes place in the stomach region because of the presence of gastric lipase, but majority of digestion takes place in the small intestine. Do you know why? Because of the presence of pancreatic lipase. So, here we have discussed the three digestion, that is carbohydrate, protein and fat, and how does they digest in the different part of our alimentary canal. If you have not watched the last two video of my channel, do watch, and if you are new to my channel, do subscribe, like and comment. Thank you. Have a nice day.